And now I will discuss the information for these next two topics. So, topic 3 is about specializations of cell surfaces, and topic 4 is about cell differentiation, histogenesis, and embryogenesis. So, cell cycle includes the different stages of life of a cell. So, a new cell develops as a result of division of mitotic division of mother cells. So once again, we discuss the cell cycle. Cell cycle is a cycle of life of a cell. So mother cell divides via mitosis, and new two daughter cells appear. Then this is the mitosis stage of the life circle, which is here. Then goes next stage G1, then stage S, then G2, and once again mitosis. So this will be the cycle. But a daughter cell can exit this cycle of division and start to differentiate. So next there will be differentiation when undifferentiated cells start to differentiate, and it means that they will develop some specific features for performing their next specific functions. And next we will have differentiated cells or mature cells. They cannot divide anymore, but they are specialized for performing of their specific function. Next, these mature cells senescence, and finally they die and are replaced by new cells. So, what means specialization? Specialization means that the cell has some specific organelles or some specific features of ultrastructure that are needed to perform the specific function of a cell. These stages of life of one cell are called differon. So differon is the totality of cells of one type on all stages of differentiation. It means that in most of tissues we will have cells on different stages of differentiation. For example, in our skin that covers our body, we will see the cells which are called keratinocytes on all of these stages of development, starting from stem cells or cambial cells that are dividing and making new cells, up to aging and dying cells, and also post-cellular structures, squames of skin. So all of them taken all together will be called differon of keratinocytes. Differon is the totality of cells of one type on all stages of differentiation. Here you can see the diagram of developing formed elements of blood, and in this picture you can see that all of the blood cells, such as lymphocytes, neutrophils, basophils, macrophages, platelets, and erythrocytes, they all develop from one stem cell, which is called hematopoietic stem cell. So it is the first stage of development for all of them. So, for example, differon of erythrocytes will include hematopoietic stem cell, multipotent stem cell, then this common myeloid progenitor cell, and finally erythrocyte. So it will be different of erythrocytes, including all stages of development of erythrocytes, starting from hematopoietic stem cell. Different of T lymphocytes will include also hematopoietic stem cell, multipotent stem cell, then common lymphoid progenitor, and finally T lymphocyte, like this. During differentiation, what happens with a stem cell, which is Campbell cell? It loses the ability to divide and changes to a mature form. And what happens? First of all, the ratio between the volume of cell nucleus and volume of cytoplasm changes. The undifferentiated or stem cell has higher nuclear to cytoplasm ratio. It means that volume of cytoplasm is very small because no specific organelles are present in high amount in this cell. So such undifferentiated cell has all of organelles, but they all are few. And then when this cell undifferentiated starts to differentiate, the ratio between the volume of nucleus and volume of cytoplasm decreases. It means that volume of cytoplasm increases. The volume of nucleus can decrease, but not always. It may stay the same, but always, almost always, at least, volume of cytoplasm increases during the differentiation of a cell. Because a cell, when it differentiates, it develops a lot of some specific organelles which will be needed to perform the specific function of such a cell. And these organelles will take some volume of cytoplasm. And so volume of cytoplasm increases. And this is for blood cells. This is the neutrophil with smaller nucleus than in undifferentiated form and higher ratio of cytoplasm because there is a lot of specific granules with specific substances in a cytoplasm of a mature neutrophil. The same is for nerve cell. This is undifferentiated Campbell stem cell, and this is the mature neuron.
Excuse me, Professor.
Yes?
I have a question. At the blood cells, in a two-step, is a smaller nucleus?
At first, the ratio between nucleus and cytoplasm is 1 to 1.
Yes.
And in differentiated form, the volume of cytoplasm increases 2 times, so that ratio between the volume of nucleus and volume of cytoplasm becomes 1 to 2. And in
Okay.
some also in some cases, like in the example of neutrophils, the volume of nucleus also decreases, and also it leads to decrease of this ratio.
Okay. Thank you so much.
So in most of the cases, this ratio between nucleus and cytoplasm in differentiated cells decreases as a result of growing of cytoplasm. And we will see that most of the stem cells have fewer volume of cytoplasm than mature cells. Mature cells have a lot of specific organelles in cytoplasm, so that volume of cytoplasm is much higher than in a stem cell. And now we discussed the cell surface specializations. It means that first of all, a cell has some specific surfaces, and second, that these surfaces may have some specific features for performing specific functions. So we will discuss them using the epithelial cell as an example. The epithelial cell has three kinds of surfaces. The top one is called apical surface. It is here. The basal lower surface is called basal surface, and side surfaces are called lateral surfaces. And a cell can have different specializations of all these surfaces. The apical surface can have cilia. This is one cilium showed in this picture, or it may have microvilli here, one villus, many microvilli, or some kind of elongated microvilli, which are called stereocilia in specific sensory cells, or kinocilia also in sensory cells. Not all of the cells have them all. It means that the presence of some specific cell surface specialization depends on the function of a cell. It means that some cells may have only cilia upon their apical surface, while other cells have only microvilli upon their apical surface, or maybe a cell has no cell surface specializations. So it depends on functions of a cell. Next, a lateral surface includes cell junctions, because here you can see that adjacent cells come close together and they are attached to each other with some specific contacts, junctions between adjacent cells. And on the basal surface we can have anchoring junctions, which are hemidesmosomes, and basal invaginations or basal labyrinth. Here you can see the functions of these cell surface specializations. So cilia, this is one cilium. Cilia, they can beat together. It means that they very fast bend to one and other side. They make it together, and while they do so, they move the fluid that covers the surface of such cells. So the beating of a lot of cilia helps to move the fluid which covers the apical surface of such a cell. For example, in the cells of trachea, the epithelium of trachea. Next, microvilli. These are for increasing the cell surface for absorption function. So microvilli are present, for example, in intestinal epithelium cells, where these cells are needed for absorption of food. Stereocilia and kinocilia provide reception of some stimuli. Cell junctions provide attachment and communication between adjacent cells. And on the basal surface, basal invaginations or basal labyrinth is for active transport of ions into the cell or out of the cell. And anchoring junctions, which are hemidesmosomes, are for attachment of a cell to the basement membrane. So, starting from apical cell surface specialization. So here in this picture you can see parts of two adjacent cells. One is here, second is here, left, and we are looking at their apical surfaces. So here we can see the cilia. Cilia are high, long protrusions or evaginations of cell membrane and cytoplasm with a complex of microtubules which are inside them. And this complex of microtubules inside the cilia is called axoneme. The function is to move fluids that cover the surface of a cell. The example is the ciliated cells in trachea that move mucus with dust particles and bacteria that stick into this mucus outside from lungs toward the pharynx. Microvilli, these are shorter than cilia. They are also protruders of cell membrane and cytoplasm, but with microfilaments inside them. In cilia, we have microtubules. In microvilli, we have microfilaments. And microvilli are for increasing the cell surface for absorption. Cilia is plural. When we have many cilia, one cilium. Microvilli, when we have a lot of them, and one microvillus. Here on the left picture you can see the scanning electron microphotograph of the apical surface of tracheal epithelium. So here you can see that some cells have long cilia upon their surface, while other cells have shorter microvilli. And here in this picture you can see the picture of what the cilia are doing. So these are cilia, and they bend back and forth all together, and they so move the fluid, which is yellowish here. This is the mucus which covers the surface of this epithelium, and these bluish, these are virus particles, and greenish, these are bacteria from air which we inhalate during breath. So these dust particles, bacteria and viruses stick to sticky mucus covering the surface of the epithelium of trachea, and cilia beat together and move this mucus with trapped viruses and bacteria upwards from lungs towards pharynx. And finally in pharynx this mucus is swallowed or coughed out, etc. Here in the upper right picture you can see the transmissional electron microscopy showing some cilia with darker microtubules inside them and shorter microvilli of two adjacent cells. And on the right bottom cell picture you can see cross sections of cilia. These are larger circles with cross-sected microtubules inside them. So this is axonema with cross-sected microtubules inside them. So these are sections of cilia, and smaller sections of microvilli. No microtubules, only microfilaments inside them. Now, the structure of cilia. One cilium, here it is shown also in this electron microscopy picture. It has the axoneme part, which grows from the cell. It is located outside of the cell, and it has microtubules inside it. These microtubules are arranged in such complex form so that these microtubules are paired. They go two in pairs. So we have one pair of microtubules in the center and 9 of the pairs at the periphery. So these red circles are cross-sected microtubules, and these pairs are called doublets of microtubules. And one of the microtubule in each pair has so-called dynein arms. This dynein protein is a protein which helps to move along the microtubule. So this dynein protein uses ATP energy to move one pair of microtubules along adjacent one. And when it happens, all of axon M kind of bend to one direction when dynein proteins act. When they stop to perform their function, then axon M goes to its previous state. And it happens very, very fast. Next, psyllium has basal body and striated rootlet parts. These parts are anchored inside the cytoplasm of a cell. And this is how they look in cross-section. So basal body has triplets of microtubules, and striated rootlet has also triplets of microtubules and a central cylinder. The striated rootlet is attached inside the cytoplasm of a cell to terminal web. If you remember, terminal web is the network of elements of cytoskeleton of a cell, and one of the function of this terminal web is to attach the cilia, if a cell has cilia. In this picture you can see transverse or cross sections of some cilia. So you can see that the psyllium has a membrane, cytoplasm, and doublets of microtubules, 9 of them at periphery and 1 pair in the center. So function is to move fluids. Next, microvilli. Microvilli also are the protrusions of cell membrane and cytoplasm, and they have actin microfilaments inside them. These are actin microfilaments which are attached also to the terminal web inside the cytoplasm of the cell. The terminal web consists of myosin microfilaments, intermediate filaments, and microtubules. And actin microfilaments inside the microvillus and myosin filaments inside terminal web, they can move along each other like they do in muscle cells, where we have actin and myosin myofilaments, which are for contraction. Almost the same we have here. So these actin microfilaments interact with myosin microfilaments to attach to each other and also to regulate the height of the microvillus. So the height or the length of microvillus can be changed during its function. Next, inside microvillus, the actin microfilaments are attached together with the help of cross-linker proteins such as fimbrine, epsin, and villin. And also they are attached to the cell membrane with the specific mini-myosin molecules. Actually, you should learn and remember all these proteins which you see in this presentation, and also this will be in your task in workbook, in your album. Also here you can see that the membrane of the microvilli is covered with glycocalyx, and as you remember from the previous lecture, glycocalyx in this case serves here it is, also serves for membrane digestion of food, because we are talking about exactly the cells of the intestinal epithelium, which is for absorption. So some of these molecules of glycocalyx are actually the enzymes attached with their one end to cell membrane and free another end, and these enzymes are for breaking down the food particles, exactly the dimers into monomers, and monomers then go through the cell membrane inside the cytoplasm of microvilli and then to the cytoplasm of the cell. If we make the cross-section of microvilli, this is what we will see: transverse section, membrane covered with glycocalyx. Inside we have cytoplasm with actin microfilaments. Each actin microfilament here is cross-sected and looks like one small black dot. When a cell has a lot of microvilli, like you can see here, 1, 2, 3, 4 cells, each of them has a lot of microvilli, then we call this structure a brush border, a lot of microvilli covering the cell surface. And this is how this brush border looks like in our light microscopy picture. We cannot see each single microvillus, but when they are multiple, they make this line covering the apical surface of the cells, which is called brush border, and it is a specific feature of the intestinal epithelium. Brush border is the structure which is composed of microvilli. So if a cell has a lot of microvilli, they look like a line covering the apical surface of the cell. So brush border consists of a lot of microvilli. We cannot see each of them, but they all together make this specific brush border. Yes. Next, lateral surface specialization. Lateral means side surface of the cell. Here we have two adjacent cells. They interact together, forming the junctions or contacts between each other. And these are specific contacts located at different levels. So here is the top surface or apical surface of the cells. Here is basal surface of the cells. If we are talking about epithelium cells, like we see in this picture, when we go from the apical surface towards the basal surface, we will have these junctions in a particular order. So the first one, always the closest to the apical surface, here first, will be tight junction. Tight it is called because here the membranes of adjacent cells come close together and are pinned together very tight, and this tight junction prevents flow of any substances between the cells. It kind of seals the intercellular space, the space between the cells, so nothing now can pass between the cells. Next will be adherence junction or belt desmosomes. It is for junction for attachment, mechanical attachment of the cells. Next will be desmosome or macular adherence, and next can go lateral invaginations and gap junctions or nexuses. Here in this diagram you can see the classification of these junctions according to their function. So first, occluding junctions. They are tight junctions, showed here. So they are first when we go from top to bottom between the cells. Tight junctions. They seal two cells together and prevent leakage of solids and particles between the cells. For example, for the cells of intestinal epithelium or for the cells of epithelium of stomach, if you know stomach is filled with acid and also enzymes which can break down all the proteins, and we don't want this acidic content of stomach to go through the epithelium of stomach to underlying tissues. So tight junctions between the cells prevent the leakage of this acid, if we are talking about the stomach, between the cells to underlying tissues. Next, mechanical attachments or junctions are lateral interdigitations, like we have seen here. It is when two membranes of two cells make these finger-like protrusions in two sides. It is to increase the area of contact between the cells, and it is also a type of mechanical attachment. And also we have mechanical attachments such as spot desmosomes or just desmosome, was here desmosome, belt desmosome or zonular adherens, which is here adherens junction or belt desmosome. These junctions can have different names. Some of these names are specific English names, some of them are Latin names. We give you all the names because in different books or maybe in tests you can meet any of this name. So just for you to know what does it mean. And hemidesmosome. Hemidesmosome, it is here. It is not between the two cells, but it is the contact between the cell and underlying so-called basement membrane to attach the cell to basement membrane. And finally, communicating junctions. They are gap junctions or nexuses. It is here, gap junction or nexus between the cells. And synapse. But synapse is the specific junction between the neuron cells in nervous tissue, which is shown here, where the vesicles with neurotransmitter are opened into the cleft between the pre- and post-synaptic membranes, and neurotransmitter acts upon postsynaptic membrane, causing the generation of action potential. But we will not talk about synapse now, we need only gap junction. Here you can see how these specific cell junctions look like when we see them on our electron microscopy pictures. These are pictures that you will see during colloquium and test, so look upon them very carefully. So in the picture on the left you can see a part of top parts of two adjacent epithelial cells of the intestine, because they have microvilli. These are microvilli upon their apical surface. Some of them look like that they are not attached to the membrane, like this one, for example. It looks like this microvillus is not attached to the cell, but it's just the result of sectioning. Every microvillus is growing from the cell. Here you can see the actin microfilaments attached to the terminal web here. Here is the terminal web. And now we are talking about this area, the area of the junctions between the membranes of these two adjacent cells. So first will go the tight junction, occluding junction, which seals the cells together. Next will go the zonal adherens or belt desmosome. It will be always on the level of the terminal web, because the terminal web consists mainly of microfilaments, and these microfilaments are the parts of this particular junction. Next will go macular adherens or desmosome. And here you can see also gap junction. Zonal adherens has no fibrillar structures here, just two membranes which are attached together very closely. And it is the first contact when we go from the top part of the cell to its bottom part. Zonal adherens has some fibrillar material, and this thickening, more dark looking, and it's always located at the level of the terminal web, because the microfilaments of terminal web are parts of this contact. Next, desmosome looks like this. It has mustache-like looking because of thick intermediate filaments, which are parts of this particular junction. And gap junction looks like tight junction. It has no fibrillar material here, but it is not at the top, not closely to the apical surface of the cells, at some other level. In this picture here you have lower magnification of the same cells, and here microvilli also. This is the level of terminal web, more dark gray color you can see here. So here will be tight junction. At the level of terminal web we have belt desmosome or zonal adherens or adherens junction. And next, these are interdigitations, finger-like protrusions. No desmosomes and gap junctions are seen in this particular picture. The same as here. So two adjacent cells, first tight junction. This is how it really looks like. It is when two membranes come close together and they are sealed together with specific proteins that come through both membranes from cytoplasm of one cell to cytoplasm of second cell and pin two membranes very close together. Next, belt desmosome. It's for mechanical attachment of adjacent cells. It has microfilaments in its structure, like this. Desmosome, also for mechanical attachment of adjacent cells. It has thicker intermediate filaments in its structure. And nexus, it is here, it is for communication between the cytoplasm of two adjacent cells, because this contact, this junction, is composed of the proteins which make the ion channels between the cytoplasm of one cell and second cell, and some very small molecules and ions can pass through these ion channels from cytoplasm of one cell to cytoplasm of second cell. So gap junctions are for exchange between the cells with some ions and small molecules. Here the same, tight junction, intermediate junction or zonal adherens, desmosome, and gap junction. And you can see here the major general structure of them. So tight junction is composed of specific proteins which pin one membrane to second one. Zonal adherens is composed of the attachment plaque and actin filaments and the cell adhesion proteins. between the membranes of the cells. And desmosome also has these three components: attachment plaque, elements of cytoskeleton, but not microfilaments, but intermediate filaments this time, and cell adhesion proteins. Attachment plaque is the structure composed of some specific proteins, and it serves to attach cell adhesion proteins to intermediate filaments here, or to microfilaments in the case of the intermediate junction. And cell adhesion proteins, they are transmembrane proteins. One end of such molecule is inside the cell, it passes through membrane, and its second end is located outside of the cell, between the cell. And the first end of this molecule, which is inside the cytoplasm of the cell, is attached to the attachment plaque. And here you can see how the cell adhesion proteins from cell 1 are attached to the same cell adhesion proteins from cell 2. And here they are attached to each other, making this junction between the cells. And gap junction, or nexus, is composed of the ion channels that enable passage of ions from cytoplasm of one cell through this channel, which is called connexon, to second cell. Now, about all these junctions, once again in details, actually you will need all the names of these proteins, because you will have the task in your workbook, and also it will be asked during the colloquium. So look at these particular proteins. So tight junction locates at the top. Here are microvilli of two adjacent cells. Tight junction. Two membranes come close together, and they are sealed with transmembrane proteins, occludin and claudin. Occludin or claudin. And nothing can pass between the membranes of these cells, between the cells. Next, spot desmosome, or just desmosome. No, second should go the intermediate filament, or belt desmosome. It is called belt because it goes like a belt at the perimeter of the cell. It is second contact. It is composed of three components: cell adhesion proteins, which are called cadherins. One end of such protein is inside of cell, it goes through membrane, and second end goes outside. And these three ends attach to each other between two membranes of adjacent cells. Second component, attachment plaque. It is composed of the proteins which are called catenin, vinculin, and alpha-actinins. These are actin-binding proteins. The function of attachment plaque, once again, is to attach cadherins, cell adhesion proteins, to intracellular, which means inside the cell, actin filaments, elements of cytoskeleton, and cytoskeleton microfilaments, which go along the membrane of the cell. So here it is in the picture. Upwards will be tight junction, and here is our belt desmosome. This is the level of the terminal web, more grayish looking area. So between the cells, between the membranes of two cells, there will be the cadherins, cell adhesion proteins. They are not very well seen here. Two membranes here, and attachment plaque composed of some actin-binding proteins attached to cell membrane from inside of each of the cells, and actin filaments are from the terminal web. And this is how the location of actin filaments looks like if we use them as the marker for the cells. They are red here, and cadherins are yellowish here, so they are located between the cells. Next will be spot desmosome or just desmosome. It is composed of the same three structures: cell adhesion proteins, transmembrane proteins, cadherins. This is the class name for these proteins, but they have some specific names: desmocholine and desmoglein. These are the names of these cell adhesion proteins. They are attached to the attachment plaque, red in this picture. Attachment plaque here is composed of proteins desmoplakin and placoglobins, and also to this attachment plaque the intermediate filaments are attached. Intermediate filaments in epithelial tissues are composed of protein keratin. And this is how this desmosome looks like in our electron microscopy pictures. So here are two adjacent membranes of adjacent cells. Between them we will see cell adhesion proteins, cadherins, desmocholine and desmoglein. Next, two membranes, and they look very thick because the attachment plaques are attached to each cell membrane from inside of each cells. So these thickenings are actually the attachment plaques, and these are the intermediate filaments of protein keratin. Next, nexus or gap junction is for communication between the cells, and it is composed of transmembrane ion channels. Each of this channel, which is called connexon, it has two halves. One half is in the membrane of one cell and second half in the membrane of second cell. Each of them is composed of 6 proteins, and these channels can be opened like in picture above, and or closed as on the picture below. So when they are opened, then ions or some small molecules can easily pass from cytoplasm of one cell to another cell. And this is how this nexus looks like in our electron microscopy pictures. So here you can see just two membranes of cells attached very closely together. No thickenings like attachment plaques, or no intermediate or microfilaments, nothing is seen here, only two membranes. The elements of cytoskeleton are involved in all of the cell junctions except of gap junctions, as you can see here. So spot desmosomes has in its structure intermediate filaments, red in this picture. They go from one desmosome to another one, etc. The cell membrane of each cell is covered with these spot desmosomes, so they are attached to intermediate filaments, and intermediate filaments so go from one cell membrane of the cell to another one. Intermediate junction or belt desmosome has microfilaments in its structure, and microfilaments also make the terminal web here. Tight junction zonal occludens also is attached to microfilaments, Only gap junctions are not attached to any elements of cytoskeleton. And now basal cell surface specializations, we have two of them. Hemidesmosomes for attachment of the cell to the basement membrane, and basal labyrinth. Here they are. On the right, you can see the hemidesmosome, which function is to attach a cell to the basement membrane, and on the left, basal labyrinth is for active transport of ions from the cell or out of the cell. And now we talk about each of them in details. So basal labyrinth. Basal labyrinth is composed of two elements. First, mitochondria. Here they are. They are ordinary mitochondria with lamellar cristae, which serve for ATP molecules production. And second component of basal labyrinth is basal folds. They are folds of cell membrane of its basal part. So the membrane of a cell makes these infoldings inside the cytoplasm of the cell. And in cytoplasm, between these folds, the mitochondria are located. The function of this specialization basal labyrinth is active transport of ions from cell to the underlying tissue or into the cell. This is how it looks like in electron microscopy pictures, and if we use light microscopy, this specific structure will look like basal striations. Striations means, like you can see, some lines. In this case, these lines will be located at the basal part of the cell, below its nucleus. So here is actually the light microscopy picture, the section of the duct of the salivary gland. It consists, it's a tube consisting of the cells, and here is one of such cells, and you can see here that its basal portion is turned into basal labyrinth with folds of membrane and a lot of mitochondria. Now, why membrane makes folds here? To increase cell surface area, to increase the amount of integral proteins that perform functions of transport of ions. So to increase the amount of ion channels or ion pumps. Ion pumps, unlike ion channels, use ATP molecules to make it possible for ions to pass through membrane, and they help the active transport, the transport of ions against the concentration gradient. And because of the presence of a lot of ion pumps, pumps are ATPases. It means that they break down ATP and use the energy of ATP to perform their functions. So ion pumps need ATP molecules, and this is why such a cell has a lot of mitochondria, because we need a lot of ATP energy for ion pumps. So if a cell performs a function of active transport of ions against the concentration gradient, then it will have folds of membrane with increased amount of ion pumps and a lot of mitochondria. So basal labyrinth is present only in those cells that perform this specific function, active transport of ions against concentration gradient. Examples of such cells are the cells of specific striated ducts of salivary glands, and also the cells of nephrons inside kidneys, and some other cells. And now structure of hemidesmosome. Hemidesmosome is for attachment of a cell to basement membrane. Now the epithelial cell stays upon basement membrane. Now we should look at the basement membrane. If we return back here, in these pictures, basement membrane is shown as a yellowish line. So, as you remember, tissue consists of cells and extracellular matrix. The basement membrane is a component of extracellular matrix produced by the cells, and it has very specific structure. It is rather hard, and it helps cells to be attached to this basement membrane so that they can make one layer or one row of the cells standing on the same level, standing upon the same basement membrane. All the epithelial cells have at least one layer of cells attached to this basement membrane. So the presence of basement membrane is the feature of epithelial cells, epithelial tissues. The components of the basal membrane are three layers. These are Latin names: lamina lucida, lamina densa, and lamina reticulata. Lamina means thin layer. Lucida means light. Densa means dense, and reticulata means reticulate or net looking. In this picture you can see the border between the cell is above, this is the basal portion of the cell, and everything which is below, this is the basement membrane upon which this cell is situated. So this is the membrane of this cell, and here should be the cytoplasm of the cell. Just below the cell membrane, there starts the first layer of basement membrane, which is called lamina lucida, which means light layer, and it is really light looking. And if we look at real transmissional electron microscopy picture, you can see it how it really looks like. So here the same. This is the cytoplasm of a cell, and here is its membrane, and everything which is below, this is the basement membrane. It is very high magnification. So cell membrane, it is here, thin black line, and below it whitish lighter line. This is lamina lucida of the basement membrane, just below the cell membrane. Next we'll go lamina densa. It is densely packed and it looks darker in electron microscopy. So here it is, this grayish line. This is lamina densa. And next below it, reticular lamina, or lamina reticulata, which means looking like a meshwork of some fibers. And now the components of these layers of basement membrane are those. So lamina lucida, which is just below the cell membrane. Cell membrane in this picture is this green line, and below it we have lamina lucida. It's composed of laminins, which serve to attach the cell, its proteins integrins, which are transmembrane proteins, the free end of which is located just here in lamina lucida, and laminin binds these integrins to underlying the lamina densa. So below it we have lamina densa. It is composed of collagen type 4. Collagen is the protein from which collagen fibers are made. So we are located outside of the cell, so we are in extracellular matrix, and here in basement membrane, in lamina densa, we have collagen 4 proteins which make very dense network. It is really very dense, so it is dark looking in electron microscopy picture. And collagen is bound with laminin to integrins from the cell. And below the lamina densa we have lamina reticulata, which has collagen fibrils, which consist of collagen type 1 and type 3. So these lines here going in different directions, they are collagen fibrils, composed of collagen type 1 and type 3. And they are attached to lamina densa with the help of anchoring fibrils, made up of collagen 7. These are them. The same is here. So in lamina lucida we have laminin, in lamina densa collagen type 4, and in reticular lamina we have collagen fibrils. Here they are grayish, which are attached to lamina densa with the help of anchoring fibrils of collagen 7. You will need it also for your workbook, all of these names of proteins. And now, how the cell actually is attached to the basement membrane is that the hemidesmosome helps to attach the cell to the basement membrane. Hemidesmosome is called so because it looks like a half of a desmosome. Hemi means 1/2. It really resembles 1/2 of a desmosome. Desmosome, as you remember, is the junction between two adjacent cells. So hemidesmosome also has 3 components: extracellular proteins, integrins, here they are green here, transmembrane proteins, one end of them is located inside the cell attached to the attaching plaque, and second free end is located in lamina lucida of the basal of the basement membrane. Next, attachment plaque and intermediate filaments. Attachment plaque, it consists of protein plectin, and intermediate filaments consist of protein keratin. The same is here, structure of hemidesmosome. In this picture you can see a portion of the basal part of the cell. It is here. Here I show you the cell membrane of a cell. It is thin black line, and below it, everything below it is the basement membrane. Below the basement membrane, the connective tissue starts. So basement membrane consists of lamina lucida, whitish line, lamina densa, dark gray line, and lamina reticulata with fibrils of collagen just below it. This is the cytoplasm of the cell. So hemidesmosome has 3 components: cell adhesion proteins. They will be located in lamina lucida. Cell adhesion proteins, transmembrane proteins in integrins. One end of them is inside the cell, and second end is inside basement membrane, inside lamina lucida. A second component of hemidesmosome, attachment plaque. Here it is. Its protein is plectin. the attachment plaque is shown here with red arrows. It looks like thickening of a membrane of a cell, more dark and thick line here. And a third component of hemidesmosome are the intermediate filaments of cytoskeleton. They are composed of protein keratin in epithelial tissues, and these intermediate filaments make here in this cell such bundles, very well seen. These are bundles of intermediate filaments inside the cell. So we have discussed all the material from for topic 3, and now we go to next topic, topic 4, which is about the cell differentiation, histogenesis, and embryogenesis. So we started our lecture with this picture, cell cycle, which includes mitotic cycle, during which cell divide and proliferate. Proliferation means cell dividing and making more cells with the help of mitosis. It is when one mother cell divides into two daughter cells, which are equal to their mother cells. And then some of daughter cells can also divide, and some can exit this mitotic cycle and go to differentiation. They start to differentiate to mature form. They become specialized cells with their specific functions. They cannot proliferate anymore. They perform their function for some period of time, and then they grow old, and finally they die. In this picture you can see this for a neuron. So first it was neuroblast, the stem cell for a neuron. Stem cell is the cell which is non-differentiated. It doesn't perform specific function. It just divides and makes new cells. And it is called stem or Campbell cell. Next it grows, becomes specialized cell, neurocyte, neuron in our case. It has specific features, specific structures such as long processes, axons and dendrites, synapses. And finally it grows older and dies. And according to the ability of the cell populations to renew itself, to make more new cells, the cell populations are subdivided into 3: renewing, growing, and static or stable. Renewing means that this population of cells has always the stem cells in it. It means that in any time we have stem cells that can divide and make us more cells. It is, for example, for the epithelium, for epithelium of our skin, for example, where we have a lot of stem cells that always divide and make new cells. New cells grow, get older, finally they die, become squames of the skin, and are replaced by new cells. Next, growing type is when we have no or few Campbell cells, stem cells in the population, and the cells are not renewing themselves normally, but when it is needed, for example, during some damage to tissue, they can return to mitotic cycle and divide again and make new cells again. Or in this population we can have some amount of stem cells that will divide only when it is needed. Not always, but only in some cases, for example, in the case of damage to tissue. And finally, static or stable populations have no stem cells and no ability to make new cells, no ability to proliferate, to divide. And it means that in the case of damage to such tissues, they cannot be regenerated, replaced, and healed with restorage of their function. It is true for neurons. It means that in our brain, for example, we have no stem cells for neurons, so no new neurons can be born. And also for cardiomyocytes, the cells of our heart. They also cannot interface is when the cell doesn't divide. It has the nucleus with nucleolus, centrioles, and DNA is in the form of U and heterochromatin. Next, prophase, when the cell um, starts its mitotic cycle, um, mitotic spindle appears, the DNA is now becomes visible because the chromosomes are formed from the chromatin. Um, the membrane of nucleus disappears. Next, metaphase is when chromosomes are visible. They are located at the central plane of the cell, and they are attached with microtubules to the mitotic spindle. The centrioles go to poles of the cell. Next, anaphase is when the chromatids start to move towards opposite centers, to opposite centrioles. And telophase is when two newly born daughter cells start to divide from each other. And late telophase is when the two nuclei are now covered with nuclear envelope, and the cytoplasm of two cells also divides. So here you can see the mitotic phases in animal cells. So the same as in a plant cell. I will send you the picture shows the showing the stages in each case. Here, during cell division, in some cases, we will have the polyploid nuclei. For example, it is true the telophase doesn't happen, and we have the cell with doubled amount of DNA, of chromosomes, and DNA in its nucleus, and now it is a tetraploid, not diploid as other cells. And it is needed to increase the functional activity of some cells. Or in the case of aberrant mitosis, it means that it is not normal, something went bad. We can have, as a result, tumor cells with not normal amount of DNA, and such cells can make tumors. You have the example of mitosis in plant cell as a using the root of onion as an example. So here are plant cells. In some of them you can see chromosomes, so they are dividing cells. Other cells are in interphase, they are not dividing. In them you can see the nuclei with nucleolus. And if you see chromosomes, it means that the cell divides. For example, here is the anaphase, late anaphase, metaphase, and in some cases telophase, not seen in this picture. Here, this is our histological preparation of section of the skin with sebaceous gland. This is the so-called thin skin with hair. So it is the section of some surface of our skin with hair. We have hair almost everywhere except some areas such as palms of our arms and some others. So here we have a part of skin with hair. Hair is here. In this picture, it is here. It is anchored deep inside our skin, and its shaft is over the surface of the skin. And so-called sebaceous glands are always present close to the hair in our skin. So here is the sebaceous gland. Sebaceous gland produces so-called sebum. It's the mixture of lipids that makes our skin looking dirty, but basically covered with lipids, which serve for protection against bacteria, against water, against air, etc. So this sebum, mixture of lipids, is produced by the cells of sebaceous gland, which are located here. They are called sebocytes, and they produce lipids, and then these lipids are released through the duct of this gland towards the surface of the hair. And finally, these lipids go to the surface of the skin. So we should look closely to the gland, to the sebaceous gland. It is composed of two parts, the secretory portion and the excretory duct, through which the lipids are removed from secretory portion, where these lipids are made by the cells called sebocytes. The same is here. We can see here secretory portion, duct is over there. Now, the sebocytes always renew. So here we will have the basal or cambial stem cells, which are dividing cells which make new sebocyte. They are located at the periphery here, just at the border between the sebaceous gland and the tissue which surrounds it, which is connective tissue with a lot of collagen fibers. So here, just at the border, there will be stem cells, basal cells. They are called basal because they lie upon the basement membrane. This thin line just at the border between the sebaceous gland and underlying connective tissue is the basement membrane upon which stem cells are located. These stem cells are the smallest cells, because all the stem cells have very few amount of cytoplasm. So here they divide and make new sebocytes, and some of them start to differentiate and become mature cells that will be here. Here the process of maturing is shown. So during the process of maturation, the stem cell produces lipids with the help of smooth endoplasmic reticulum, and these lipids are stored in lipid droplets or secretory granules, secretory vesicles filled with lipids from inside. And step by step, all the cytoplasm becomes filled with these vesicles filled with lipids. So all the cell finally is filled with these vesicles with lipids. Lipids are not stained with any stains, so the cytoplasm of such a cell looks transparent or pale with no staining. Only cell nucleus is visible. Next, when the cell is totally filled with lipids, it starts to die. So when we go closer and closer to the duct, we will see the dying cells. Dying is the process when the organelles start to disintegrate, to break down, and also nucleus starts to break down, and finally it will disappear. And finally all the cell is dead, and it is actually now just the large bubble filled with a lot of smaller vesicles filled with lipids. And this vesicle filled with lipids is then removed through excretory duct towards the surface of the skin. And here you can see the stages of death of the cell. And these stages include karyopyknosis, karyorhexis, and karyolysis. Karyon means nucleus. It's Greek for nucleus actually. And these words they are about the fate of nucleus. Pyknosis means the condensation of chromatin and the shrinkage of nucleus. It means that nucleus becomes smaller and darker, like it is, for example, here in this picture. So these closer to basement membrane, here we have young mature cells with their young mature nuclei. They are rather large and lightly stained. And here we have smaller and darker cell nuclei, and these, for example, small and dark, these are pyknotic nuclei with karyopyknosis, condensation of chromatin. Next we'll go karyorhexis. Rhexis means destruction and fragmentation of nucleus. It is when this pyknotic nucleus is breaking down into smaller particles, like you see here. For example, 1-2 particles in the place where the nucleus previously was. And finally, karyolysis. Lysis means digestion. It means that these particles of nucleus now are totally disintegrated with the help of enzymes from lysosomes. And in such cells we will have just the shadow of the nucleus in the place where this nucleus previously was, and finally no nucleus will be seen. So you will have these pictures of histological preparations of this gland, in which you should be able to find the cells during different stages of life cycle, starting from stem cells, which are located here at the periphery of the gland, just at the border between the gland itself and connective tissue outside of it. And these cells are small, with small cytoplasm. Next layer will be the layer of differentiating and mature cells, here with normal cell nuclei. And next will go cells with disintegrating nuclei, with smaller, darker, and broken nuclei. And finally here cells with no nuclei, no cytoplasm, just large bubbles filled with lipids. The same as here. Secretory portion of sebaceous gland, stem cells, basal cells at the periphery, they are here, maturing cells here, mature cells, and dying cells closer to the duct of the gland. This is the hair follicle. During cell death, we have two types actually of cell death: apoptosis and necrosis. Apoptosis is the programmed cell death. It is when it is needed for a cell to be dead, and it uses ATP molecules to perform these changes during cell death, and cell is fragmented and disintegrated into smaller particles, which are usually taken by the adjacent cells. So one cell dies, and other um, are eating the particles from this dead cell. This is true, for example, for the intestinal epithelium. It is constantly renewing, and constantly old cells die and new cells are produced by stem cells, and these dead cells can be disintegrated to smaller particles, and adjacent cells eat these particles. Necrosis is not programmed cell death. It is present, for example, as a result of some damage to tissue. And during this process, the swelling of organelles take place. All the cells become swelled, becomes larger and lighter because water goes inside the cell. And finally, the membranes are broken. The cell membrane is broken. The membranes of all organelles are broken, and the material of this dead cell is spread to all direction. This is bad for surrounding cells because the enzymes of lysosomes go out from lysosomes because membranes are ruptured, and these enzymes can damage adjacent cells. Enzymes are molecules for breaking down other molecules. They are for digestion of proteins, lipids, and sugars, and because all the cells are composed of proteins, lipids, and sugars, then they can be broken down with enzymes if the enzymes go out from lysosomes. So necrosis usually um, involve not only one cell, but a lot of cells in tissue. But apoptosis can happen just to one cell. It is not harmful to surrounding cells. And now we go to histogenesis and embryogenesis. So embryogenesis is the developing of embryo starting from the fertilization, when spermatozoon fertilizes the oocyte, and a first cell of a new organism is evolved, which is called zygote. And next zygote starts to divide via specific type of dividing, which is called cleavage, and it divides to the stage of so-called morula, which consists of several blastomeres with no cavity between them, no spaces between them. Then the cavity starts to appear inside this embryo, and now it is called blastocyst. And next, the gastrulation takes place, which consists of two phases. First phase is when the one layered embryo develops into two-layered embryo. So first phase is one layer to two layers, and second stage of gastrulation is two layers to three layers. And then the stage of organogenesis and histogenesis starts. Histo means tissues, organon means organs, and genesis means development. So development of organs and tissues starts. So, for example, neurulation takes place, which is the name for the development of neural tube, the germ of brain and spinal cord. And then organs start to develop, which are shown here. So fertilization happens inside the ovarian tube. So here is ovary. From ovary, oocyte goes out and it gets to the ovarian tube. It happens every month. And if spermatozoa come into uterus, they go towards the oocyte and they will meet inside the ovarian tube. I mean oocyte, which is egg cell, and spermatozoa. Only one spermatozoon can enter oocyte, egg cell, during fertilization, and the nucleus of spermatozoon and egg cell combine together to make new organism. First cell is called zygote. Now we have zygote, and it immediately starts to divide. And this division is called cleavage. Cleavage, unlike mitotic division, is when daughter cells are twofold smaller than mother cell. And again, each cell, which is called blastomere, divides once again, and daughter cells become smaller and smaller, and total volume of embryo doesn't increase as a result of cleavage, unlike mitotic division. Cleavage is full, asynchronous, and asymmetric, and we have first morula, when embryo consists just of blastomeres, and then when cavity starts to appear inside the embryo, now it is called blastocyst. Cleavage starts from day 1 of life of embryo till day 6, and it happens inside ovarian tube. By day 6, the embryo, it will have the form of blastocyst, should enter the uterus, where the next stage will take place. So cleavage is from day 1 to day 6. Blastomeres are not equal. Some of them are lighter, some of them are darker. Darker ones are dividing not so fast. They divide slowly, and they are larger than lighter ones, and so lighter ones divide faster, and they grow these darker ones from outside. Those which are darker and larger, they will become so-called embryoblast, and those which are smaller, they are at the periphery, they will be called trophoblast. The trophoblast cells in future will form the fetal part of placenta, the baby part of placenta. Placenta is the temporal organ for feeding of a baby inside the uterus of mother. Through placenta, the gas exchange happens between blood of mother and blood of the baby, and it happens through the blood capillaries of placenta. And placenta is composed of both maternal tissues and baby's tissues, and these baby's tissues of placenta will develop from trophoblast. All other tissues of baby itself and other temporary organs will develop from embryoblast. Tropho means feeding. Embryo means embryo. So trophoblast is for feeding temporary organ placenta. During a stage of 8 blastomeres, the compactization happens as a result of development of junctions between blastomeres. Before junctions appear, like they are here, before junctions appear, we can take one of blastomere. If we make extracorporeal fertilization, for example, and we can check the genes, the DNA inside this blastomere, if there's everything okay with DNA. For example, if mother or father have some inheritance diseases, and they do not want their baby to have such a disease, so they can go through this extracorporeal fertilization. And if everything is okay with DNA in this particular blastomere, it will be okay also inside all others, because they are all the same if we are talking about DNA. And if we take one of blastomeres, it is not harmful for others. And also, every of these blastomeres can give one baby, actually. So if we divide them all, if we detach them all from each other, we can make one baby from every one of them. If something like this happens inside the mother's uterine during uh, early development, it will give twins. So this happens inside ovarian tube, and by the day 6, the blastocyst should enter the uterine. So this is the uterine wall, and next this embryo will attach to the wall of the uterus from inside, and then it will go inside the wall. So it will kind of eat up its way through the wall of the uterus, and it will stay here. The epithelium will cover it from outside, and embryo will develop inside the tissue of the uterus, inside endometrium of the uterus. In vitro fertilization is when we take the egg cell from ovary, add some sperm to it, make an embryo, zygote, which starts to divide to perform cleavage. We take one blastomere for karyotyping. It means we check the chromosomes. We can also check DNA to be sure that everything is okay with DNA in this particular embryo. So we use the pipette to take one of blastomeres. We check it, and if everything okay, we will place this embryo into the uterus of the same woman. And next, starting from day 6, the implantation happens. Implantation is the integration of the embryo into the wall of uterus of the mother. Implantation has 2 stages: adhesion and invasion. Adhesion means attachment, invasion means going inside. Adhesion starts from the exiting of embryo from its surrounding membrane inside which it was previously, and then this embryo is attached to the surface of the uterine epithelium, which is here. It happens with the help of symplast or trophoblast, which develops now. It's the next stage of development of trophoblast. As you remember from this picture, inside we have embryoblast, outside we have trophoblast. And by day 6, trophoblast start to develop into next stage, into cytotrophoblast and symplastotrophoblast. Cyto means cells, symplasto means symplast. Symplast, you know it from the muscle fiber. Symplast is the result of fusion of cells. So cytotrophoblast cells divide and proliferate, so they make new cells, and some of them next fuse together to form symplastotrophoblast. This symplastotrophoblast has a lot of lysosomes which have enzymes in them, and they release these enzymes to break down the uterine wall, and it will cause invasion, the going of embryo inside the tissue of the uterine wall. And also symplastotrophoblast is for first stage for adhesion stage of implantation, for attachment of the embryo with the epithelium of the uterus with the help of specific receptors. So next stage, invasion, starts from day 6 up to day 12, and during this, symplastotrophoblast releases enzymes which break down the tissue of mother, and embryo goes inside this tissue. So this is what happens. Epithelium of mother's uterine, underlying connective tissue, and embryo invades inside this tissue. It goes deeper and deeper, eating its way with the help of releasing of enzymes with this symplastotrophoblast, which is shown here in green color. Also, during these days, during second week of life of embryo, from day 6-7 to day 14, provisory organs or temporary organs start to be formed. We have 4 provisory organs: amnion, chorion, yolk sac, and allantois. They are needed only during embryonic development of a baby, of the embryo. So here we have the embryo, it is here. Now in this particular picture, in this stage, embryo, future baby, consists only of 2 layers, upper and lower. Upper is called epiblast, lower is called hypoblast, so embryo is here. All other structures are temporary organs. So this bubble just above the embryo, this bubble is called amnion, and the cavity inside it is called amniotic cavity. Just below the baby we have the other bubble with its cavity. It is the yolk sac. This cavity is the yolk sac cavity. Also, this yolk sac has very small protrusion, which is called allantois, number 3, and all this large bubble, this is called chorion. Chorion, this part of which will make the placenta. So baby is here, all other organs are temporary for development inside the uterus. So how they are formed? Now by the day 6, by the beginning of implantation, the embryo, it is here now. It consists of 2 layers, epiblast and hypoblast. Next, epiblast of embryo splits with the making of cavity, which is called amniotic cavity. So this is how amnion is formed. So amnion is now here. It is the result of the split of epiblast. Next, the hypoblast cells start to divide, proliferate, and grow to both sides of embryo downwards, and they make so yolk sac. So here we have trophoblast from cyto and symplastotrophoblast. Cytotrophoblast is made of cells. This is why it is called cyto. Cyto means cells. we can see cell borders here between the cells, and symplast or trophoblast is the simplest. No cell borders are seen here, it is just multinuclear structure, which serves for release of enzymes to break down the tissue of mother's uterine to make the way for embryo to grow inside the wall of mother. And also, during this period, the development of the embryo itself takes place, which includes two stages of gastrulation. Stage 1 is called delamination, which means split. What happens? Embryoblast, blue here, it's one mass of cells, is split into epiblast and hypoblast, into two layers. So delamination is when we, from one mass of cells, have two layers of embryo. Epiblast, epi means above, and hypoblast. Hypo means below. So, as a result of delamination, which happens at day 7, we have two-layered embryo. And next, starting from day 14, we'll have second stage of gastrulation, which is called immigration. Immigration means moving of the cells to some space. So now we are looking at the embryo from above. So this is the top view of embryo. Here is its frontal end, and this is its tail end. And in its central line, in its posterior part, the cells of epiblast. We see here epiblast. We look at from the top to embryo now. The cells of epiblast start to divide very fast, and then these cells start to migrate below epiblast. So if we cut the embryo like this is what we will see. We see epiblast, it is blue, and here this line, which is called primitive streak, this is the midline through which dividing cells start to move inwards below the level of epiblast, basically between the epiblast and hypoblast. Hypoblast is yellow here. So the migration process of cells of epiblast between the epiblast and hypoblast is called immigration. Some of these cells migrate through the primitive streak, which is this line. The direction of this migration process is shown with these arrows. So the cells go from the periphery towards the midline, then they go inside between epiblast and hypoblast, and then they move towards the frontal end of the embryo and toward its side ends laterally. And this is shown here. The cells move laterally. Some of the cells go downwards and they are inserted between the cells of hypoblast. They are here. So, as a result, we will have three-layered embryo. It will be composed of three layers, 1, 2, 3. These cells, which go laterally, they will make inner layer mesoderm, and those that are inserted inside hypoblast layer, they will make the endoderm of the embryo, endoderm, and all other cells that stay in epiblast and do not go downwards, they will make ectoderm, outer layer. Also, we have the Hansen's node here it is. Unlike primitive streak, it is not like a line, it looks like a circle. Also, through this Hansen's node, cells migrate between epiblast and hypoblast, but they move direct towards the frontal end of embryo. And these cells will make first the precordial plate, so-called, and next portion of migrating cells will make the notochord. I will speak about them later. These migrating cells, as you can see them here, they are detached from the layer of epiblast, which is the epithelium with attached cells, and now the cells are detached from each other and they are moving. These detached moving cells are called mesenchymal cells or mesenchyma. They make the embryonic tissue, which is called mesenchyme. This is embryonic connective tissue. And mesenchyme, we have two types of mesenchyme, primary and secondary one. Primary develops earlier and it is seen in temporary organs. So here you see temporary organs. This lighter grayish area in structure of all of them, this is the primary mesenchyme or extra embryonic mesenchyme, because it is outside of the embryo itself. Embryo itself, it is here. It is composed now of two layers, epiblast and hypoblast. And we already have our extra embryonic or primitive primary mesenchyme. Next, during this stage, during immigration, we will have intra-embryonic mesenchyme. Mesenchyme which is inside the embryo itself. And this mesenchyme will make the tissue of baby next. Here in this picture you can see this same process in immigration starting at day 14. The arrows show us the direction of the movement of the cells. Cells of epiblast go through the epiblast layer towards this midline primitive streak, then they go between epiblast and hypoblast and move laterally and to the frontal end of embryo, making the mesoderm, and also some of the cells are inserted in the hypoblast here, and they will make the endoderm of the baby. Those cells that migrate through the Hansen's node, this rounded structure area, they will make first precordial plate, small portion of cells, and next portion will make notochord. And we are looking at the embryonic disc, at the embryo itself, which is here. Embryo now consists of two layers, epi and hypoblast, and it is covered from above with amnion, and below it we have yolk sac, and everything is inside chorion, and all of it is inside the mother's uterine. The same as here. Arrows indicate direction of movement of the cells of mesoderm and cells of endoderm, and here will be the cells migrating through the Hansen's node, which will make the precordial plate and notochord. So the same as here. And now as a result of second stage of gastrulation, if we make the section of the embryo, this is cross section if we cut it like this, and this is sagittal section if we cut it like this along it. This is what we will see. Result of the second stage of gastrulation is the appearance of 3 layers of embryo: ectoderm, mesoderm, and endoderm. The same is here: ectoderm, mesoderm, and endoderm. The embryo itself is here. It is covered with amnion, and below it is yolk sac. So, blastocyst gave us embryoblast and trophoblast. Trophoblast develops into cyto and symplastotrophoblast. In this picture, we can see these cyto and symplastotrophoblast. They are for invasion stage of embryo, and they are for formation of placenta next. Embryoblast, which is shown here in blue color, it will give first epiblast and hypoblast of the baby. Here blue in color epiblast, yellow hypoblast. Epiblast will give us ectoderm, mesoderm, and endoderm. Epiblast, here migration of its cells starts. Some of the cells make mesoderm, some of the cells make endoderm, and the cells that do not migrate make ectoderm. So all of the 3 layers of embryo, ectoderm, mesoderm, and endoderm, develop from epiblast as a result of migration. Next, ectoderm is actually embryonic and extra-embryonic or amniotic. So embryonic ectoderm, it is here, and the wall of amnion cavity is made of extra-embryonic or amniotic ectoderm. Mesoderm is also extra-embryonic and embryonic. It is the same as mesenchyme. So extra-embryonic mesenchyme is shown here in red color. It is the part of all the extra-embryonic organs. This is chorion, consisting of cytotrophoblast, symplastotrophoblast, and extra-embryonic mesoderm. Amnion consists of amniotic epithelium or amniotic and ectoderm and extra-embryonic mesoderm, and yolk sac. It consists of extra-embryonic endoderm and extra-embryonic mesoderm. No intra-embryonic mesoderm is seen here yet, because the immigration process didn't start yet in this picture. It starts here. And now here we will have the intra-embryonic or just embryonic mesoderm. Hypoblast will give extra-embryonic endoderm, which is the wall of yolk sac, these yellow cells in the wall of yolk sac. Next, when we finally have 3 layers in our embryo, ectoderm, mesoderm, and endoderm, so this is the embryo itself. Above it is amnion, below it is yolk sac. So now we have 3 layers in embryo, and now histogenesis and organogenesis starts. So what we see here, the mesoderm, which is red in color here, starts to develop, and it develops into so-called somites, legs of somites or somite stalk, and the lateral plate mesoderm, which is here. 2 layers of lateral plate mesoderm, upper and lower. So this is development of mesoderm. It develops into somite stalk, and the coelomic epithelium or lateral plate mesoderm, consisting of 2 layers. Also, during the same period of time, the ectoderm develops into ectoderm itself, which will make skin of our body, and the neural tube, which will give nervous system. So this process is called neurulation. So central area of ectoderm starts to invaginate, and then it closes with the formation of neural tube, and here also neural crest, not shown here. And also during the same period of time, the embryo starts to change its form. So first the embryo is flat. It is in the form of embryonic disc, as we see it here. We are looking from above. It is like a flat disc consisting of two layers, ectoderm, mesoderm, and endoderm. And now it starts to bend like this. The lateral folds start to appear, and these folds will grow to each other, and finally the embryo will close and it will resemble the tube. So first flat embryo will become tube-like embryo, elongated like the worm. So neurulation, the development of neural tissue, neural system, begins at day 17 up to day 25. So notochord somehow acts upon the ectoderm and makes this ectoderm to become a first neural plate that next invaginates into underlying mesenchyme with the formation of neural groove, and next it closes with the result of formation of neural tube and some cells that are not included, not into neural tube nor into epidermis of ectoderm, and they stay in a form of neural crest or ganglionic plate. And these cells of neural crest shown here, neural tube is here, neural crest is here, neural tube is here, neural crest is here, between ectoderm and neural tube. And the cells of neural crest go downwards, ventrally to the ventral surface of the future embryo, and laterally along the surface of the future skin of embryo, along the surface of ectoderm. And the cells of neural crest develop into a lot of different types of cells and tissues, which are listed here in this slide, and this will be also the task in your album, where you should name the derivatives of neural crest. So you should take them from this slide. So neural crest will develop of neurons and nerves of ganglia. Ganglia are groups of neurons outside of brain and spinal cord. So in our body we have neurons, nerve cells, not only in brain and in spinal cord, but also outside of them in peripheral nervous system. For example, we have neurons also in our intestines, in our gut, which is shown here. Next, calcitonin producing cells of thyroid gland here, bones and cartilage of skull, derm and adipose tissue of the face and ventral neck area, ciliary muscle of the eye, number of striated muscle in the head, and some other structures. And those cells that are migrating along the future skin, along the ectoderm, will develop into melanocytes of our skin, the pigmented cells. And others develop, which migrate ventrally, they develop into neurons, supporting glial cells of the peripheral nervous system, sensory ganglion cells, endocrine cells of adrenal medulla, in adrenal gland, prevertebral plexuses, also neurons. So these are the derivatives of neural crest. The neural tube will develop into brain and spinal cord. So here you can see the same process, starting from migration of gastrulation, second stage of gastrulation, organogenesis, neurulation, and these are the pictures also that you will see in the questions for colloquium, in the tests, or also these are the sections of embryo after gastrulation took place. So this is the cross section of embryo, like this. We see 3 layers: ectoderm, mesoderm, and endoderm, and centrally located notochord. As you remember, notochord develops from the cells migrating through the Hansen's node towards the frontal part of embryo. Notochord. Notochord helps neural tube to develop from ectoderm, and then it disintegrates, and it is present finally only in the intervertebral discs. Next, the same but sagittal section, like this. So bluish is ectoderm, yellow endoderm, and here in the caudal part of embryo, posterior part of embryo, we have mesoderm, and here in frontal part we have notochord, because the section was made like this through notochord, and before notochord we have precaudal plate. The same is here. Here the ectoderm is kind of removed from the underlying mesoderm and endoderm. So what we see here, ectoderm, the neural tube just starts to develop, so the process of neurulation takes place here. This is the same stage that is here. So neural groove is formed, and here it is also formed. Neural tube is not closed yet. And below, what we see here, endoderm is whitish, it's the lower layer, and upon it the middle layer is in the middle notochord, and before it precaudal plate. Like in this picture, notochord, it is here, and before it precaudal plate. In this picture we can see only notochord, because the section was made like this, at the level of notochord, but the precaudal plate is not included in this section. So notochord and precaudal plate, these are central organs, and the mesoderm, which consists of somites. These are bunches of tissue, some portions of tissue, which are located in pairs along the notochord. They develop from rostral part to caudal part, from the frontal to end part of embryo, and they develop into 35 segments, pairs of somites. These are somite stalks, and lateral plate mesoderm, which should be here, is not included in this picture. So somites are these areas, larger areas of mesoderm. These smaller are somite stalks, and lateral plate mesoderm, it is here. It consists of two layers, upper and lower. So organogenesis and histogenesis. What we see here, neurulation is ended, so neural tube is closed now. Ectoderm covers the embryo from outside. Endoderm, it is here. After lateral folding will end, the endoderm will be closed, and it will make the embryonic gut, which is the primitive intestine of embryo. This is notochord, and these are somites, somite stalks, and lateral plate mesoderm. Somite, stalk, which other name is nephrogonotome. In this name, the names of the structures, the organs that will develop from it, are encoded. So this area will give rise to nephro. Nephro is for kidney, for urinary system, and gono is for gonads, which are testes and ovaries, for reproductive system. So nephrogonotome will give rise to organs of urinary and reproductive system. And lateral plate mesoderm with two layers: inner layer or visceral layer or splanchnoplura, and outer layer, which is parietal or somatopleura. So this is the real picture of histological section of the embryo at this stage. So what we see here, here will be ectoderm. It covers the embryo from outside. Here will be endoderm. Between them we see mesoderm with its somite stalk or nephrogonotome, and lateral plate mesoderm consisting of two layers: parietal or somatopleura and visceral or splanchnoplura. Here is notochord, and this is neural tube. Next, the somite starts to develop into three parts. The outer part will be called dermatome, the middle portion will be called myotome, and innermost portion will be called sclerotome. So first, the somite was the portion of tissue, and now it is developed into three parts. Here is the same structure in real histological preparation of the embryo. Ectoderm is here, neural tube is here, below it notochord, and in this area we have the somite, and now it is developed into outermost dermatome, the middle portion myotome, and innermost portion sclerotome. The dermatome portion, outermost, it is closest to the ectoderm, which will give rise to the epithelium of our skin, and dermatome will give rise to dermal layer of the skin. Our skin consists of epithelium and underlying connective tissue, which is called dermis. So ectoderm will give rise to epithelium, and dermatome will give rise to dermis of skin. Next, myotome. Myo is for muscles. Myotome will give rise to skeletal muscles, all the muscle fibers, symplasts that you know already. And sclerotome will give rise to skeleton tissues, for bone and cartilage tissues. Mesenchyme is the form of developing migrating cells. So here also, as you can see, the cells of sclerotome are present in the form of mesenchyme. They are not attached together like the cells of neural tube or in ectoderm, for example. They are detached from each other, like in this picture. They are with some processes, and they are migrating. So they are in form of mesenchyme, which is the connective tissue of embryo. The cells with thin, long processes and extracellular matrix around them. So when the skeleton develops, for example, the bones of our arms, the cells from sclerotome should migrate to the area of embryo where this bone of our arm will develop. and then they stop there and start to develop into bone cell in the bone. So while they are migrating, they are in form of mesenchyme. Now, the derivatives of mesoderm. Mesoderm consists of notochord, somites, nephrogonotomes, and lateral plate mesoderm. Notochord disintegrates and stays only as a small portions in intervertebral discs. Somites are developed into dermatomes, myotomes, and sclerotomes. Dermatomes will give us the dermis of the skin. Myotomes will develop into skeletal muscles. Sclerotomes will give us bone and cartilage tissue, and also connective tissue, blood vessels, and smooth muscle. Nephrogonotomes will give us the excretory organs, the urinary systems, and gonads, which are the reproductive organs of the reproductive system. Splanchnotomes, or lateral plate mesoderm, consisting of two layers, visceral and parietal, will give rise to mesothelium, and visceral layer will give rise also to myocardium and epicardium layers of the heart. Lateral folds shown here change the shape of the embryo. The embryo first was flat, and now it will be closed with the centrally located gut developed from the endoderm, the body cavity coelom, and outermost layer ectoderm that will cover the embryo from all the sides. So here are our histological preparations, the organogenesis and histogenesis. We have the sections of embryo at three stages. The first picture, this is the embryo at the second stage of gastrulation, immigration. This area in the middle, this invagination of tissue, it is actually the primitive streak or primary streak through which the cells, migrating cells of epiblast, detach from epiblast and migrate between the epi and hypoblast. Hypoblast is below. Second stage is the stage of organogenesis, because here we have the neural tube, the developing mesoderm. Now it consists of somites, somite stalk, and the lateral plate mesoderm. And now here we can see the embryo at the stage of lateral folding. These are lateral folds that will make the embryo to become worm-like. So here the somite already developed into three parts: dermatome, myotome, and sclerotome. In nephrogonotome you can see some cross-sected tubules. These tubules are the tubules of primary kidney. So kidneys are developing here, and also some tubules for reproductive system. This is the outer and here the inner layers of lateral plate mesoderm. They both will make the flattened epithelium, which covers the organs which are located in cavities and lines from within all the body cavities. For example, our intestines, our guts, and our stomach, intestines are located inside our body cavity, and this cavity is lined with mesothelium, and all the organs, all intestines and stomach also are covered with mesothelium. The same as for pericardiac cardiac cavity inside which the heart is located. So the cavity is lined with mesothelium and heart is covered with mesothelium, and between these layers of mesothelium there is this serous fluid which lowers the friction, which ensures the beating of heart without the friction. So the mesothelium which lines the body cavity will be made from the parietal layer of lateral plate mesoderm and mesothelium which cover the organs inside this cavity. It will be developed from the visceral layer of lateral plate mesoderm. Here, where gut is developing, this is the endoderm. Actually, this is the embryonic endoderm, and when these folds will meet together, this part will give us the embryonic gut. All other portion of this endoderm will stay outside of the embryo when these folds will fuse together, and so all this area, the rest area of endoderm, will give us the extra embryonic endoderm. The same is for other tissues of embryo. So outermost here it is the ectoderm. It is embryonic ectoderm. It will cover the body of embryo. And this is extra embryonic endoderm. It stays outside of the embryo and it takes place in formation of amnion. Mesoderm, lateral plate mesoderm. This will give the mesoderm of embryo, and this will give extra embryonic mesoderm. The same is for visceral layer. Before the level of the fold, it will be intra-embryonic mesoderm, and outside of this level of the fold, it will be the extra embryonic. Extra means outside, and intra means inside. Next, self-aid and determination and differentiation. What these words mean. So, during the cleavage of the zygote, the first cell of the embryo, when the blastomeres are formed, all of these blastomeres are totipotent. So here you have the totipotency. Potency means the ability to develop into different ways. Toti means any. So it means that each blastomere can develop into any cell of intra or extra embryonic tissue, and it means that actually you can take one blastomere and grow a baby of it with all its intra and extra embryonic organs. So any cell can develop from blastomere, and it means totipotency. Toti means any, every. Next, during gastrulation stage of embryo itself, when we finally have three layers, ectoderm, endoderm, and mesoderm, as a result of second stage of gastrulation, cell potencies are specified by embryonic layer, and now they are not totipotent, but they are poly or pluripotent. Pluri or poly means many potencies, many ways of possible development, but not toti. It means that the cells of mesoderm can develop only into the cells which develop from mesoderm. From mesoderm, as you know, develop mesothelium and also dermis of skin, and from myotome skeletal muscles, and from sclerotome the bones and cartilage tissues. So if you take the cell of mesoderm, you can grow a skeletal muscle, for example, from it, or a cartilage cell, or a cell of connective tissue of dermis. Any cell that can actually develop from mesoderm, but not the keratinocyte of skin, which develops from ectoderm. So the amount of ways of development is lower than in blastomeres, so it is called restriction of potencies. Restrictions means cutting off the potencies. So the cells of mesoderm cannot develop into the cells of ectoderm, and vice versa. Labile determination and transdetermination also is possible. Next, ectoderm develop into neuroectoderm and integumentary ectoderm. Neuroectoderm gives rise to the structures of nervous system, neural tube, and neural crest, and integumentary means skin. Ectoderm will give us the skin and nails, hair, and etc. So organogenesis happens now, and here the cells now finally the cells will be unipotent. It means that the neuroblast will be able to develop only into neuron, not into the cell of the intestine or into muscle fiber, only to neuron. The determination is stable and also cannot be changed, so canalization of development happens. So development goes into only one direction. But in our adult organism, we also have in some areas stem cells that are pluripotent. Not all the cells of our body are unipotent. Some of them are polypotent, means that they can develop into different ways. For example, the hematopoietic stem cell, which is located in our red bone marrow in bones, which can give rise to any cell of our blood. So this hematopoietic stem cell is pluripotent, and such cell which is pluripotent, which is stem cell, but it is located in adult body, it is called adult or resident stem cell. Embryonic stem cells can be taken from embryo, and if we cultivate it, we can grow any cell type from it. But from the stem cell which is resident, from the stem cell hematopoietic, we can grow only cells of blood, not neurons and not intestinal cells. Here we use the example of the skin to make the examples of differons. The cell differon, or the same histogenetic series, consists of the cells of the same type but on different stages of development. So here these lightly bluish, these are the cells of the main differon of the skin epithelium, which are called keratinocytes. So here at the basement layer, lying upon the basement membrane, you can see the dividing cell. Here proliferation takes place, so here we have stem cells, which next are going upwards to the surface of the skin. So from stem cells, first they will become differentiating cells, next fully mature cells, and next dying cells. Death of the cells leads to formation of squames of the skin. So here we have squames, and also here we have in the same tissue of the epidermis of the skin other cell differons. For example, the macrophage, which is called Langerhans cell, it serves for protection against bacteria here. It is not developed from keratinocyte stem cell, it develops from the hematopoietic stem cell in red bone marrow. It comes here from blood vessels, goes out, and goes here inside the layer of the skin. Also, we have here pigment cell, melanocyte, which gives the skin its color. Merkel cell, which is the cell for attachment sensitivity. So these are all other cell types, or as we name them, they are other differons of epidermis, except of the main one, the differon of keratinocytes. So here we can see the pictures of our histological preparations. This is called the primitive streak in a chick embryo in the cross-section. So this is a cross-section of the embryonic disc, and in its central area you can see this area with the invagination. And this invagination is basically called the primitive streak. This is the cross-section of primitive streak through which cells are migrating in this direction between epiblast and hypoblast. The same is here, the primitive streak through which cells are migrating between epi and hypoblast. Epiblast is here, hypoblast is here. The same is here. Second stage, somites notochord and neural tube of chick embryo also in cross-section. Here we have already 3 layers: ectoderm, endoderm, and between them mesoderm. Mesoderm consists of somite, somite stalk, and lateral plate mesoderm. Also we have here central organs, neural tube, and notochord. And finally lateral and amniotic folds in chick embryo, with these lateral folds which grow towards each other, and all the structures that we were discussing. So here is the same neural tube, notochord. The somites developed into dermatome, myotome, and sclerotome. In nephrogonotomes, the tubules of nephrons and organs of the reproductive system are already developing, and these are the parietal and visceral layers of lateral plate mesoderm. This area will be the body cavity, for example the abdominal cavity inside which the intestine is located. So intestine itself will develop from this area. So as a result of fusion of these lateral folds, this area will be closed. So this will be the wall of the intestine. Here the endoderm, embryonic endoderm, will give the innermost layer of intestine. The mesenchyme here will give all other layers, and outermost layer of the intestine wall will be the mesothelium, developing from the visceral layer of splanchnotom. The intestine will be located inside the abdominal cavity, which develops from this cavity, which is called here coelom. And coelom, the abdominal cavity, will be lined with mesothelium, developing from parietal layer of splanchnotom, this one. And the same as here, neural tube, notochord, somites, dermatome, myotome, sclerotome, dorsal aorta already develops, inside which we see the first cells of blood. So this is all. I will send you the materials for you to study, to fill your album, the translations of album. So if you have any questions, I will be glad to answer them. And I hope to see you in class.
Thank you, Doctor.
So thank you everyone for attention. Have a good day.
You too. Goodbye.